Antibiotic resistance in marine predatory fishes. Our group has myself doing the introduction and conclusion, Krista Schiller doing the methods, Cade Richards reporting on the results, and Tyler Cross working on the discussion. The study that we focused on was evidence of antibiotic resistance in free-swimming top-level marine predatory fishes, which was written by Jason K. Blackburn and his associates. The idea was that antibiotics were created in the 1940s, and since then bacteria have shown a great capability to develop many various resistant strains to nearly all forms of antibiotics. This presents a major problem to both human and veterinary medicine. In addition to this problem, there's been very limited research for the impacts of this on marine animals, as well as how the impact on marine animals may affect people. And the goal of this main study was to analyze the trends of antibiotic resistance in various species of sharks and other types of fishes, and observing whether or not this may change in various different locations. The hypothesis of the experiment was that antibiotic resistance, first of all, would be able to be detected in marine predatory fishes, and that the prevalence of this resistance will decrease the further that an animal is found from shore, indicating that the closer they are to humans and pollution would indicate a higher rate of antibiotic resistance. There was also the idea that sharks of the same species will have resistance rates that vary by location, indicating that their environment would have an impact on the rate more than their species. Antibiotic resistance will also vary between species in the same geographic location, depending on genetics, biology, and the animal's age. Our next speaker will introduce the methods of this study, Krista.
There were six study sites used in this study. Belize, the Florida Keys, Tomlier Bay, Louisiana, an offshore oil rig in Louisiana, a second offshore oil rig also located in Louisiana, and Massachusetts. And different populations or different species of fish were tested at each of these sites. Belize and the Florida Keys, they both had nurse sharks. Tomlier Bay, Louisiana, they had bull sharks, black-tip sharks, and lemon sharks. At the first offshore oil rig, spinner sharks were tested. At the second offshore oil rig, redfish were tested, and in Massachusetts, dogfish were tested. And this is just a graph that summarizes parts of the study. The first column on the left has to deal with the site. The second column is the geographic location. The third is the ecological setting. The fourth is the different species that were collected at each location. The fifth is the sampling gear, like what was used to catch all the different fish. And the sixth is the number of bacteria isolates. Bacteria samples were collected by swabbing the cloaca or distal anus with a sterile rayon tip applicator. And then these swabs were inserted 5 to 6 centimeters and rotated several times. And they were put into transport media and sat on wet ice and transported to Louisiana State University School of Veterinary Medicine for processing. Once the bacteria samples were at the Louisiana State University Medical Center, bacteria samples were inoculated on 5% blood agar plates by using the swab applicator from the transport media and were incubated at 37 degrees Celsius for 24 hours. Presumptive organisms were described by using gram stain and colony morphology. Gram-negative isolates were also cultured on McConkey agar for 24 hours at 37 degrees Celsius to see if they fermented lactose. And isolates were characterized and described by grain scan and morphology. Bacteria were tested for antibiotic resistance by using the Kirby-Bauer disc diffusion technique. And 13 drugs were evaluated in this study. In this chart lists all the 13 drugs, the dosage, and just the abbreviations that were used throughout the study. The plates were then incubated at 37 degrees Celsius for 24 to 48 hours. And after the samples had been incubated, the diameter of the zone of inhibition around each disc was measured and recorded as resistant or susceptible. And total resistance and multi-drug resistance were also calculated throughout this study. Total resistance was calculated as the total number of isolates in each population with resistance to at least one drug divided by the total number of isolates in each population. Multi-drug resistance was calculated as the total number of isolates with resistance to more than one drug in each population divided by the total number of samples in that population. Different tests were performed in the study. The Pearson chi-square tests were calculated to quantify potential interpopulation differences between spatially explicit groups of animals. This test was also used to test for interspecific differences between Louisiana offshore sharks and offshore redfish. The Fisher exact test was used to test for interpopulation total resistance differences between sexes in bull sharks and to test for interspecific differences between nerve shark populations because some cell counts were fewer than 5. And then a power analysis was performed for any comparison that was found to not be significant, and then a p-value of less than 0.5 was used to determine significance.
The results consisted of 130 isolates from 6 populations characterized by gram stain and morphology. These 130 isolates consisted of 77 gram-negative rods, 14 gram-positive rods, 38 gram-positive cocci, and 1 gram-negative cocci. 12 viable bacteria were isolated from 8 nurse sharks from the Belize population, 5 females with a mean length of 152 centimeters. There were 3 males with a mean length of 129 centimeters, and all of these sharks were classified as juveniles based on their size. Gram-negative rods showed resistance to 8 of the tested drugs, and gram-positive cocci showed resistance to 6 of the tested drugs. 29 viable bacteria were isolated from 7 nurse sharks from the Florida Keys. 1 shark was a female, the other 6 were males. All sharks again were juveniles based on size. Gram-negative rods showed resistance to 8 of the tested drugs, and gram-positive cocci showed resistance to 6 of the tested drugs. 46 viable bacteria were isolated from 30 sharks in Louisiana coastal waters. 10 of these were female bull sharks with a mean length of 112 centimeters. 16 were male bull sharks with a mean length of 96 centimeters. There were 2 female blacktip sharks with lengths of 64 centimeters and 70 centimeters, 1 male blacktip shark, and 1 female lemon shark. All sharks were juveniles, again based on their size. Gram-negative rods showed resistance to 6 of the tested drugs. Gram-positive cocci showed resistance to 4 of the tested drugs, and gram-positive rods showed resistance to 7 of the tested drugs. 23 viable bacteria were isolated from 14 sharks in Louisiana offshore waters, 35 kilometers southwest of the Mississippi River, and at an oil rig located 10 kilometers offshore. There were 4 female spinner sharks, 8 male spinner sharks, 2 female blacktip sharks, with ages varying between juvenile and adult. Gram-negative rods showed resistance to 7 of the tested drugs. Gram-negative cocci showed resistance to 1 of the tested drugs. 12 viable bacteria were isolated from 7 redfish collected at an oil rig in the Louisiana offshore waters. All redfish were classified as adults with mean lengths of 105 centimeters. Gram-negative rods showed resistance to 6 of the tested drugs. Gram-positive rods showed resistance to 4 of the tested drugs. Gram-positive cocci showed resistance to 2 of the tested drugs. 8 viable bacteria were isolated from 3 smooth dogfish collected from Vineyard Haven Harbor. All 3 dogfish were classified as adults. Gram-negative rods showed resistance to 3 of the drugs tested, and gram-positive cocci showed resistance to 1 of the drugs tested. The final results showed that redfish had significantly higher total resistance than spinner sharks, with a p-value of 0.0016 and 1 degree of freedom. No significant difference in total resistance between Louisiana inshore sharks and offshore sharks. There was a p-value of 0.152 and a degree of freedom of 1. There was no significant difference in total resistance between male and female bull sharks in Louisiana coastal waters, with a p-value of 0.9343 and a degree of freedom of 1. And there was no significant difference in total resistance between nurse sharks from Belize and Florida. The p-value was 0.3407 with 1 degree of freedom.
So the primary objective of this study was to determine if bacteria from the cloaca or distal anus exhibited drug resistance. Based on our results, or their results, antibiotic-resistant bacteria were present among all populations in every geographic location. The resistance is seen in demersal and pelagic food webs in South America. These refer to the bottom-dwelling fish and sharks and their trophic levels. Some of these are prevalent among fish harvested for human consumption. The low-level fish used for consumption have been seen with antibiotic resistance. There's residue in the tissue seen, and the fate of the residue may increase selection pressure, which can cause the evolution of the bacterial strains and their prevalence in antibiotic resistance. Correlated with this study is the predation of crab species and other low-level fish species among large apex predators in the degrel and pelagic food webs. And this correlates with the positive correlation between resistance and drugs to shark populations tested. The cause of antibiotic resistance is a mutation in the bacterial's genome or horizontal gene transfer, which is very prevalent among bacterial species. Horizontal gene transfer is just the transfer of genetic information or a plasmid between two or more bacterial specimens through a sex pilus or the collection of DNA absorbed. Another characteristic of this bacterial is there might be in-strength resistance among different strands of bacteria. This resistance is determined by their gram-negative stained bacteria and is prevalent in their phenotype with multi-drug resistance. 13 drugs were tested and resistance was identified for all of the drugs and at least some of the isolates tested. Some of these include chloroamphenicol, which is prevalent in the initial stages of trying to treat infections, along with ciprofloxacin and gentamicin, which is also prevalent in the initial treatment for infection. Multi-drug resistance of the 13 drugs tested was shown, and samples took from all species, no matter where their geographical location they were found, either inshore or offshore. So why is there a rise in antibiotic resistance? Antibiotic resistance has been linked to different studies, such as them in dolphins and other species. So raw sewage leaking in these areas where the shark and fish species are living is prevalent. High percentage of sewage was found in inshore locations where the nerf sharks and blackfin sharks were tested. There's also a speculation about close contact among humans in the nerf sharks in Belize, but this requires further experimentation. Another is the Mississippi runoff in the United States. There's a lot of agricultural drugs and genetic material that is washed down this river, and once it hits the ocean levels, is incorporated into the pelagic and demersal food webs, which can cause this.
This study was able to confirm the first part of its hypothesis that antibiotic resistance was in fact present in marine predatory fishes, from multiple taxa and multiple geographic locations. For example, the nurse sharks in the Florida Keys, and Belize locations had varying resistance rates, and this is partially because of a high prevalence with subpopulations in the Florida Keys compared to other locations. This could be a result of the Florida sharks being in an area with failing septic facilities, which would cause them to be in more contact with human waste, which could transfer over antibiotic resistance through bacteria in the human waste. The redfish also showed a higher total resistance than most of the other shark species tested, and this was mostly because of a bias in the study where these animals just happened to be at an older age, which meant that they had been exposed to these bacteria over a longer time period of their life, and that they had been in an environment longer that had a higher amount of the bacteria present. However, there also were other parts of the hypothesis that the study was unable to prove and would require further testing. There was no significant difference in total resistance between Louisiana inshore and offshore sharks found, and there was no significant difference found between varying genders of the same species in this study. This could partially be due to a limited sample size and not having a wide enough of sample population in each location or of each gender. There were several strengths and weaknesses to this experiment. The animals observed were long-living and slow-growing, which enhanced their exposure to the bacteria in their environment and gave them a higher potential of developing the antibiotic resistance. We also used a wide range of geographic locations that were included and studied for this experiment, and several different species were analyzed in different locations to show that it was something species-specific and not necessarily location-specific, and varieties of species in general were included throughout the experiment. There are also weaknesses, though. There was a limited sample size, and sharks have a very vast and diverse habitat, which made it hard for them to be tracked and studied, which limits any progress that can be made with long-term experiments to see how much the antibiotic resistance would change over time. And even though the animals were long-living, the specimens that just happened to be in the sample population were usually juvenile, which meant that they hadn't been exposed to the bacteria in the environment as long as an adult animal would have. Looking forward for future studies, they should have a larger sample size for each location, and they should focus on animals that are older so that they've had a longer exposure and will have more of amount of the resistance build up likely, and more specifically analyze data from each location so that more generalizations can be drawn from each location. And this information could easily be linked to health risks from fisheries because people are consuming the meat that contains this antibiotic resistance, which could cause problems with human medicine and also several different health risks, including zoonotic diseases. Methods to evaluate the genetics behind antibiotic resistance could also be tested to see if it's something that's passed on or if it's prevalence in a certain bloodline of a type of species or if it is just species specific. And quantification of nurse shark populations by location could be used to determine a higher resistance. Our references were available to us, and it was by Blackburn and his associates in the article.